Okay. All right. So thank you for joining me today for my genetics honors video submission. My Nobel Prize winner topic was the 2007 Nobel Prize in Physiology and Medicine. Um, this was awarded jointly to Mario R. Capecchi, Sir Martin J. Evans, and Oliver Smithies for their discoveries introducing specific gene modifications in mice by using embryonic stem cells. Now, we've all seen the movies and shows with labs where they have mice in little cages to test on. The house mouse has been scientists' experimental animal of choice for the last 2 decades. Now, today, genetically altered mice are an essential component to the experimental toolkit with thousands of varieties contributing to research in laboratories around the world. Their existence stems from discoveries made in the 1980s by this year's Nobel's laureates in physiology or medicine. So, quick introduction. I am Riley. I'm a second year at NC State. I like science, anything spooky. As you can tell, I took a little bit of liberty in choosing the theme of the slideshow just to make it a little more entertaining for those of you watching. So thank you and follow along with me throughout this. Looking at our laureates, I want to talk about them first and just some of their biographies. Just kind of overall summary of what they did. There was Mario Capecchi and Oliver Smithies, who were both seeking ways to specifically alter mammalian genome. Capecchi was going about it with the view of inserting new genes into cells. And Smithies he was hoping to correct genetic defeats or defects that led to diseases. Now, working against the background of skepticism, they independently discovered that they could use a natural mechanism revealed decades before by Joshua Lederberg in bacteria to introduce short sequences of manipulated DNA into chromosomes of mammalian cells growing in the laboratory. The technique allowed them to target individual genes with exquisite precision, producing the genetic alteration they sought, but only at the cellular level. Now, luckily for them, Martin Evans was studying embryonic stem cells, and he was developing the necessary vehicle for taking such gene manipulation that Smithies and Capecchi were using to make it inheritable and to actually use it long term. Now, by combining the two, they were able to use the embryonic stem cells and then inject those cells into fertilized mice and eggs or mouse eggs. This made it possible to rear mice with discrete genetic modifications that would be inherited to their offspring. These were called knockout mice for the ability to knock out unwanted genes, or in some cases, knock in mice to do the opposite and put genes into the mouse that they wanted. Now, that was just kind of the overview of what they did together, but let's look at their lives separately leading up to this big discovery. Mario Capecchi defined his life as a great example of how, despite the complete absence of an early nurturing environment, the intrinsic drive to make a difference in our world is not easily quenched, and that given an opportunity, early handicaps can be overcome and dreams achieved. And you're going to learn a bit why he had this view on life. Now, Mario Capecchi was born in Verona, Italy on October 6, 1937. Horace John Mulaney once said 1930 bad. World War II was raging. There was fascism all throughout the country. Mario's own life started in a place of poverty. His mother, Lucy Ramberg, was a poet, and his father, Luciano Capecchi, an officer in the Italian Air Force. His mother opted not to marry him, and probably for good reason considering the military at the time, but this left Mario with an estranged relationship with his father. Now, later, his mother's work would catch the attention of German authorities. Anticipating her arrest, she would sell all of her possessions, donate them to a local family that owned a farm so that they would take Mario in and raise him while she was gone. Now, he lived on the farm for about a year before the funds ran out, and after that, he was left on his own. At 4.5, he set out and lived on the streets for 4 years. He would join gangs of homeless children, maybe the occasional orphanage, but he was completely on his own throughout that time. And eventually, he was very lucky and was able to be reunited with his mother. And from there, they were able to move to America. And although she lived until she was 82 years old, his mother never psychologically recovered from her wartime experiences. And now, at the total age of 8 years old, Mario Capecci moves in with his aunt and uncle in America, with his mother, Edward and Sarah Ramberg. Edward was his mother's youngest brother and was a brilliant physicist. Sarah and Edward took on the challenge of converting Capecci into a productive human being. He had received little to no formal education on the street. He knew no English, and from the very first day he got to America, he started school. Um, it was actually Edward and Sarah who took on his education and really taught him what it meant to make his own destiny in life. He realized that whatever happened to him would be up to him and the work he put into it. So he went to Southampton Public School, where his aunt took on the task of teaching him to read from the start. And he was able to attend an outstanding high school with enthusiastic and competent and caring teachers who were able to pour into him. And from there, he was able to attend Antioch College, where he took his studies head on. um, Kaveci carried the change of making this world a better, more and equitable place for all people. His motivation, that motivation stayed with him throughout his entire education and later his career. He took every mathematics, physics, chemistry course at Antioch, including, and I've never heard of these courses, so give me a second to pronounce them: Boolean algebra and topology, electrodynamics, and then physical chemistry. He found physics and mathematics to be intellectually satisfying, but it became apparent that what he was learning came from the past. And he was looking for a science where the individual investigator had a more intimate, hands-on involvement with the experiments. Now, molecular biology was a field that was just being born in the late 1950s, and it was a entirely new breed of scientists, but it really spoke and captivated Mario Capecci, and that is what he devoted his life to from there. And moving on to his work in that field, he did many things, including made some discoveries with DNA and how it carries organisms' genomes and determining their vital processes. He felt the ability to artificially alter DNA opens the way to both new scientific understanding and new treatment methods of various illnesses. And in the early 1980s, Mario Capecci discovered that sections of DNA that had been inserted into the cell of nuclei of mammals could be incorporated into the cell's genome, all leading to his work with smithies that successfully achieved specific modifications in the genomes of mice by inactivating specific genes to determine their function. All right, and let me take a breather here. We're going to be talking about Sir Martin J. Evans next. Similar to Mario, he was born in January 1941 in his grandparents' house in Roadborough, in near Stroud in Gloucestershire, where his mother had fled to escape the bombing in London. He was also born into a period of war and had a couple of bumps in the road growing up. He, in his very early years, contracted mumps and led to a stay in the hospital, and for the next 18 months, suffered a lot of just about every available childhood infection there was at the time. He hardly got to school and spent long periods in bed with various books and toys, but this only filled his desire to learn. One Christmas, he received an electrical experimental set which the main item was an induction coil to generate high voltage shocks. He built the Tesla coil and other static electric gadgets. From there, he found one of his greatest amateur hobbies, beginning with a chemistry set. He was able to learn basic chemistry on his own. He bought supplementary chemicals from chemist shops. He made all sorts of dangerous experiments, but he considered his most dangerous as a child to be an attempt to make a large batch of urea formaldehyde resin in the fireplace of his bedroom, which took off and overheated, nearly suffocating him in the process. He felt that his personal exploration as a child was really able to foster and just allow him to be uninhibited in his desire to learn and without proper safety precautions that we just don't really allow children today. And then from there, he was able to move on to school. And as he got into middle school, he found that he was getting to learn about all of the topics that he had read in his books and experiment with, and got to learn the mechanisms behind what was really going along, as well as got to learn the proper pronunciation for a lot of the words he had only read in the books. Now, in prep school, he was taught biology by Reverend Toller, a wonderful man who used his classes to give Evans foundations in plants and animal science and cosmology. And from there, he was able to move on and win a major scholarship to Christ College in Cambridge, where he embarked on zoology, botany, and chemistry with the clear intention of following specialization in chemistry. Now, sadly, he had a setback and was not able to take his finals that you can imagine he was working hard for. And due to this fact, he wasn't able to get the grant nor the internship post-grad position that he wanted. And I could, as college students, we can relate in the idea that that was probably a scary time for him and feeling like there was nowhere else to go. But he was able to secure a research position as an assistant to Dr. Elizabeth Duchar at University of College in London, which he later looked back on as a fortunate choice. She was an excellent supervisor who encouraged but didn't direct, a policy which he later adapted for himself in teaching graduate students. And it did result in him taking a longer time to complete his PhD, but he had the time to experiment, to develop techniques. He learned skills he would have never had the time if he had focused entirely on his PhD to learn. And while viewed coldly, many might say he wasted a lot of time, but he just felt like he was never under the pressure that so many of us are today in the field of science. And from there, he was able to develop a really just significant career for science as a whole. His ambition was to isolate developmentally controlled mRNA. At the time, none of the cloning tools or probes we use today were available. So all he could look at were double reciprocally labeled profiles of polyribosomes and messenger RNA from dissected blastula and gastrula in ectoderm by sucrose density gradient centrifugation and RNA by agarose electrophoresis. That took a minute to come out. And in 1990, he moved from leading a personal research group in a scientifically buzzing institute to heading a large and newly formulated school of biosciences in Cardiff University. He was able to facilitate scientific careers on a wider scale and to help lead and develop newly re-energized Welsh University into the 21st century. It was hard, but overall it was extremely rewarding. Now, our final laureate was Oliver Smithies, who he was born in England, June 23rd, 1925, alongside his twin brother Roger. Their father, William Smithies, was at the time working for his father, who paid him erratically. And their mother, Doris Skies, was a college graduate and taught English at Halifax Technical College, where is where she actually met their father, who was one of her students and a little bit younger than her, but they did fall in love. And they were later joined by a little sister 5 years later. Um, and she would have not survived an infected tonsil were it not for the miraculous new antibiotic drug at the time, Prontosil. And while that was great for her, Oliver had a similar incident at the age 7, except he didn't have access to the antibiotic at the time, and he spent 10 weeks bedridden with a rheumatic fever. And this illness left him with a trivial mitral valve murmur. Now, this kept him from taking place in sports in middle school and high school, but it did give him a lot of time to actually spend that time reading and making things. And sometime around 11 years old, he actually read a comic with the main character who was an inventor. And while he didn't entirely have the name for that profession at the time, he knew that's what he wanted to be when he grew up. And his father also had a love for mathematics and encouraged him and introduced him to decimals starting from a very early young age. From there, his education was able to proceed with that strong foundation of a love for learning and inventing. At just 16, he joins a supervised study of physics, chemistry, and mathematics at a more advanced level, and within a year, moved to an unsupervised study in preparation for Oxford University. Now, he chose to concentrate on physics with plans to study the subject at the university, although in the end he did choose med school and was fortunate to be rewarded a Brackenberry scholarship at Balliol College. There, he synthesized many substances and whatever else caught his fancy, including phenyl isothiocyanate and one of the worst smelling substances known to man. And he also made nitrocellulose, which is constituent of a Nobel smokeless powder, and mercury fulminate, the detonator for dynamite. So a lot like Sir Martin Evans, he had a love for explosions. And however, after several years later, he followed in his mother's footsteps in falling in love with one of his postdoctoral students, Nabua Medea. And however, after a period of trouble finding work together in Wisconsin, they moved to North Carolina, Chapel Hill, to be specific. I know that's not a school we love to talk about, but he was able to get a position in the Department of Pathology at UNC. He took a job as a full-time research professor and was able to spend even more time behind the bench getting to work hands-on. And all of his experiments using gene targeting to generate animal models of human genetic diseases have been carried out in the nurturing environment of the University of North Carolina. So um, this awarded Nobel Prize does strike a little bit close to home for us, even if it's not the best college he could have worked at in North Carolina. It is something that it's kind of cool to know that all of these men maybe maybe didn't go the conventional route for getting to where we assume that we are all going to get to med degrees and scientific discoveries. They all had roadblocks and moments where maybe they didn't think they were going to get to move forward in their careers, and they still won the Nobel Prize. So at the end of the day, it's nice to know that it's doable. Okay, welcome back. Just had to take a second to put my hair up, and we're going to continue here now with the mechanisms of what they were discovering. But I want you guys to take in mind that what was happening here really was that they weren't really doing one big experiment that led to this whole new idea. What they were is they were taking ideas and knowledge we had already discovered about genetics at the time, which wasn't a lot. And they were piecing those puzzle pieces together from other scientists to figure out how we can use it to move forward. And this was originally a two-part problem taken on by our three guys. The first part of this challenge was finding a specific gene sequence among the 3 billion base pairs that comprise the human genome and then altering it in the intended way. Now, Mario Capecci and Oliver Smithies provided a solution to this problem when they demonstrated that homologous recombination can take place in mammalian cells. Now, homologous recombination, which was already known to occur in other organisms, means that genes or gene segments within a cell can exchange information with each other. Now, they discovered this independently of each other in their own labs in their own time, just about the same time as each other. Now, on the other end of the scale, we had Evans, who was ensuring the second half of this problem was ensuring that this alteration could be passed down to the next generation. Because what is the point of creating a specific gene alteration if we cannot provide that to an entire population? Now, Martin Evans worked with embryonic stem cells, or you might hear me call them ES cells for short. This was a critical importance because these cells can develop into any cell type in the body. So what he was doing was taking a culture of cells and altering the stem cells so that when the embryo was put into a mother and the surrogate into a surrogate mother and the baby was born, they could find this altered DNA in all parts of the animal. Now, this all started for all of us. We were just fertilized eggs at one point. We found that we already possess the information required for our bodies to develop and successfully interact with our surroundings, already stored in our genes. Now, because this starts so early, is what made this all possible. The genetic material is packed into the cell nucleus, where the 2-meter long DNA helix is organized into 23 base pairs of chromosomes. One chromosome in every pair comes from each of our parents. And then when the cell divides, the chromosome in the pair can exchange genetic information through homologous recombination. Now, Capecci and Smithies had the vision that this type of genetic exchange could be used to introduce direct genetic changes in mammalian cells. It was Capecci who discovered that DNA sequences injected directly into the nucleus of mammalian cells could be incorporated into the gene of the recipient cell. Now, this inspired his idea that using this technique, he can make specific changes in any gene in a mammalian cell. Now, eager to try this out, he applied for funding from the National Institute of Health, but sadly was rejected. The grant reviewers considered it far too unlikely that the injected DNA would find its correct place in the gene pool of the target cell. Now, as it happens, Evan had a similar application rejected at about the same time. The UK Medical Research Council considered the idea overambitious. And Smithies was also wanted to make targeted changes, but his work focused mainly on an attempt to repair defects in human blood cells. He discovered that the gene for globin in the blood protein hemoglobin could be altered using homologous recombination. Now, surprisingly, the change could be brought about regardless of whether the gene was switched on or not. This result suggested that it was possible to change any of one of the genes in the gene pool at any time. So, like I said, they were working on similar projects about the same time, coming up with the same theories. And at about this time, the cell types in which Smithies and Capecci had been able to make genetic changes did not give rise to germ cells. Now, germ cells are the embryonic precursors of egg and sperm cells, gametes, and it is involved in sexual reproduction in animals. And without germ cells, it was impossible to make the genetic change that could be inherited, which is what we're going to want to see if we're trying to work on disease or in humans or any genetic alteration if we are looking for to make a population have a better chance of survival. Now, meanwhile, Evans had published results showing inheritable genetic changes just right around the same time. A series of experiments done in his laboratory using embryonic tumor cells had not provided the results he was looking for exactly, but he did discover that it was possible to grow cultures of cells with intact chromosomes from mouse embryos. He found that these embryonic stem cells can differentiate into different types of all cells in the body. Now, the cells lie dormant in various tissues and develop when there is a need for more cells of that type and can develop into any other cell. Now, for this crucial experiment, Evans used blastocytes, which are a few day old mouse embryos. He injected them with ES cells that had previously been injected with DNA from a virus. The embryos were implanted in the uterus of a surrogate mother, where they developed just like a normal baby mouse would. And when they were born, they would find viral DNA in the genome of some of the mouse pups. If you did not know, mouse pups are what they're called. I thought that was cute. The experiments demonstrated that inserted DNA could, in fact, be passed down. This groundbreaking result was published in the journal Nature in October 1986. Now, you can see here, kind of following along what that entails. Oh, I didn't mean to go forward. Um, we have our differentiated ES cell at the top. Um, it can become anything it needs to be, which could be a sperm or an egg cell, a blood cell, a nerve cell, a muscle cell. These cells are just kind of our backup for whatever we might need and are always kind of there to lay the ground for what a cell needs to look like. Now, over here we have what was going on in uh, Evan's experiment. The virus inserted to RE cells was able to take on the DNA, and then genetically modified ES cells injected into the blastocyte, which we see above is our undifferentiated ES cells. Um, then blastocytes implanted in the surrogate mother, and mouse pup was viral DNA. Out pops pup. And then we have kind of what came next as they put the results together and discovered what each other was doing. Now, Capecci had already shown that homologous recombination did occur in mammalian cells, and Smithies had used his method to introduce altered DNA into a human cell, and all this had to be done in cells and culture. Now, when Evans published his results showing the inheritance of viral DNA, all the pieces of the puzzle were on the table. It was time to make targeted genetic changes in mice that were inheritable. The work proceeded on several fronts simultaneously. Evans was the expert on ES cells. Capecci visited his lab over a Christmas holiday to learn how to handle these extraordinary cells, whereas Smithies actually received stem cells in a tube that Evans brought to him in his own pocket. Both Smithies and Capecci chose to create targeted changes in mice that code for an enzyme called hypoxanthine guanine phosphorybosytransferase. Say that 5 times fast. And or HPRT. Mutations in HPRT gene result in a rare disease, Les-Nyland syndrome, which is inherited through the X chromosome, which made it easier for them to tell when the DNA was actually inherited. Now, looking at some of the successful experiments they had, Smithies showed that he could repair a defective HPRT gene in cultured ES cells by injecting a new DNA fragment corresponding to an intact gene. The reason this particular gene was selected for the experiment was that a successful gene exchange was easy to detect. Now, only the cells that had managed to take up the functional gene could grow in a special culture medium. Now, the same year, Capecci also succeeded in inserting a specific gene sequence into the HPR gene that made the cells capable of surviving in a culture medium containing the antibiotic neomycin. Now here we can look at more of a physical demonstration here. Um, on our top right corner, we have the you can see the researcher is doing a micro injection, inserting the modified ES cells into a blastocyst. This is what it looks like in real time. And at the bottom, you can see a clear picture of what's going down. It's labeled on to the left side. The inner cell mass, blastocytes, the syringe containing ES cells, and just kind of how those work in inserting the DNA. Now, all right, now taking a step back to look at what this meant for the scientific community. What these men really did was put their knowledge together to create gene targeting with information we already had. But at the time in the 1980s, we didn't really have that much information on human genomes or really much about genetics in general. So a lot of people considered this technique useless except for in a few cases when someone who's an expert on genes targeting could use it. It was just too advanced for a lot of people. But nowadays, we have refined this so detailedly that not only can we determine at what developmental stage we need to knock in or knock out, turn on or activate a specific gene, we can actually do it to just a specific tissue in the body versus the overall whole. Now we have switched, and in the years to follow, we switched from rats to mice in experimental designs. We've actually seen close to 10,000 genes altered. We have been able to do this in things such as in our blood, our nervous systems, just about anything with the having to do with the immune system. This has just really been one of the key developments in science that has allowed us to advance so many fields, not just genetics, but physiology and molecular biology and medicine and pathology. I mean, this has really brought a new era of genetics that allowed it to move from what was just kind of a small category of biology to that was newly forming as we were studying the human genome and comparing it to other animals, but into the 21st century. I mean, at the time, we barely knew how um, things like homologous recombination would work in higher organisms, let alone in cell cultures. Uh, but of course, nowadays, this is something that we might actually learn here in the introductory course, which I just find really interesting. And it actually is kind of the reason why that they only got this Nobel Prize in 2007 for work they did in the late 80s. It was just because we didn't know how to use this fundamental groundbreaking technique they had discovered. Um, so last, I want to follow up with gene targeting and really what it is. It has allowed disease models of human pathology to be generated in a tractable mammalian system and consequently enabled experimental dissection of disease states, identification of new therapy targets, and the development of test systems for pharmacology. Finally, it is obvious that the development in the future of novel therapies to correct genetic defects in man will build on the experiments of gene modification in mice that is based on the discoveries made by Mario Capecci, Martin Evans, and Oliver Smithies. It's truly just one of those things that, like, everything we do in the future that has anything to do with correcting diseases or trying to um, fix populations to make them more suited for an environment is going to come from this. This was the foundation of really just about everything we do in genetics. And so I think it's really important to take a step back and why I wanted to spend so much time on getting to know the three men of this experiment. I really think for us college students, we can learn from them as well in the fact that while they completely almost founded a whole like century of research, everything we've really done in the last 2 decades with mice has been because of them. They all had at some point in their lives setbacks that most people now would stress out so much it would make them want to give up. Not finishing their finals, not even starting any education until like they were almost in double digits. I mean, all of them really had an experience that would at some point, I couldn't imagine feeling like I could continue and go on to the extent of winning the Nobel Prize. So that's what I want to leave you here with today. Just kind of being in wow of really everything that occurred in this experiment. And I want to thank you for listening to a pretty lengthy video. And I hope you all have a really good day. Resources.